Now, let's talk about point mutations and frameshift mutations. So here we have a simple type of mutation or DNA mutation, which are transitions and transversions, which fall under the category of point mutation. Now, what we mean by point mutations. So point mutations are changes that happen in a particular nucleotide sequence. In our DNA, each nucleotide is considered as a single point. Now, any mutation in these nucleotides are called point mutations. So in this case, we have this arrangement of nucleotides where T is always paired with A and C is always paired with G, and so forth and so on. But say for example, there is a change happen in this particular nucleotide base pairing, point mutation happens. Now, what are called transitions and what are transversions? Now, we know that there are two types of bases in the DNA. One is purine bases and the other one is pyrimidine bases. Purine bases are adenine and guanine, and the pyrimidine bases are thymine and cytosine. Adenine pairs with thymine and guanine pairs to cytosine, which is our basic rule. If it happens that a purine is replaced by another purine and a pyrimidine is replaced by another pyrimidine, the condition is called a transition type of point mutation. And if a purine is replaced by a pyrimidine and a pyrimidine is replaced by a purine, it is called a transversion mutation. So now, in our example, T pairs with A, which is a purine-pyrimidine pair. So like I've said, a purine is replaced by another purine to have a transition mutation. So here, if T is replaced by C, as what we can see in the next diagram, and A is replaced with G, which is another purine, so a pyrimidine is replaced by another pyrimidine, and the purine is replaced by another purine. That is why it is called a transition mutation. As we can recall, transition mutation is done by the replacement of the same nitrogen bases. On the other hand, transversion mutation happens when a purine is replaced by a pyrimidine or a pyrimidine is replaced by a purine. In our example, T here, which is a pyrimidine, is replaced by A, which is a purine. And A, which is a purine, is replaced by T, which is a pyrimidine. Here, replacement is done with different types of nitrogenous bases. That is why it is called transversion type of point mutation. So to recall, in transition, a purine is replaced by another purine, or a pyrimidine is replaced by another pyrimidine. And in transversion, a purine is replaced by pyrimidine, or vice versa. These mutations happen during the replication stage. Now, a point mutation can change a specific codon, resulting in an incorrect amino acid being incorporated into a protein. The result is a missense mutation. One DNA suffers a change in one base pair, causing a change in the resulting mRNA codon, which in turn codes for a different amino acid in the forming polypeptide. A nonsense mutation is a type of point mutation that results in the termination of polypeptide at the effective codon. The wild-type DNA suffers a change in one base pair that results in the appearance of a stop codon in the resulting mRNA. The stop codon causes premature termination of polypeptide synthesis. On the other hand, a frameshift mutation happens when there is an insertion or deletion which causes a destruction in the reading frame. The reading frame describes how the nucleotides are broken up into sets of codons that are each 3 nucleotides long. Each codon codes for one amino acid in a protein. You can see in the original DNA strand that all of the codons are C, A, and T, which codes for histidine. But if a single nucleotide is deleted, the reading frame shifts, causing all of the codon to change. This results to incorrect amino acids being added to the protein. Again, with the same strand of DNA, we have codons C, A, and T, which codes for histidine. This time, we will insert a single nucleotide and notice that this also shifts the reading frame. causing all the subsequent codons to change. As a result, incorrect amino acids are produced. Whenever there is a disruption in the reading frame of a gene, whether it is caused by insertion or deletion, it is a form of frameshift mutation.